Imagine a groundbreaking achievement that could change the landscape of neuroscience and mental health research forever. Researchers at Johns Hopkins University have successfully created a miniaturized version of a human brain, known as a whole brain organoid, which not only emits light, but also connects in a manner reminiscent of a real brain. But here's where it gets intriguing. This innovation could play a pivotal role in understanding neuropsychiatric conditions like autism and schizophrenia, which have traditionally proven challenging to study. Lead author Annie Cathuria, who is an assistant professor in the Department of Biomedical Engineering at Johns Hopkins, expressed the significance of their work, stating, "We've made the next generation of brain organoids." Unlike previous attempts that focused on isolated brain regions, such as the cortex, hindbrain, or midbrain, Cathuria and her team have succeeded in assembling a multi-region brain organoid, MRBO, that approximates a cohesive whole brain. Their findings, published in the esteemed journal Advanced Science, represent one of the first instances where researchers have developed an organoid that incorporates tissues from various brain regions, functioning together as a unit. This remarkable step forward could revolutionize how we investigate complex neuropsychiatric disorders that affect the entire brain, shifting the paradigm away from reliance on animal models. To construct this sophisticated organoid, Cathuria and her colleagues began by cultivating neural cells sourced from different brain regions, alongside early-stage blood vessels, in distinct laboratory dishes. They then utilized a special type of protein that acts as a biological adhesive, essentially a superglue for tissues, tying these components together. As the parts fused and began interacting, they initiated electrical activity and began behaving as a functional network. The result? A mini brain organoid that closely resembles the array of neuronal cells found in a 40-day-old human fetus. Impressively, about 80% of the various cell types typically present during the early stages of human brain development were identified within these lab-created organoids. Although significantly smaller than an actual human brain, with only 6 to 7 million neurons compared to the tens of billions found in adult brains, these organoids serve as a valuable platform for studying whole brain development. Interestingly, the team also identified the formation of an early blood-brain barrier. This crucial layer of cells plays a vital role in regulating which molecules can enter the brain, further mimicking the characteristics of a real brain environment. Cathuria highlighted an important aspect of this research. To fully grasp the intricacies of neurodevelopmental and neuropsychiatric disorders, we need to engage with human cells. It's not ethical or practical to simply ask individuals for access to their brains for study, she noted. These whole brain organoids allow us to observe the progression of disorders in real time, assess treatment efficacy, and even customize therapies for individual patients. In addition to their potential in understanding diseases, these whole brain organoids may also enhance the success rate of clinical trials for experimental drugs. Alarmingly, it's estimated that 85% to 90% of drugs fail during phase 1 clinical trials, and for those targeting neuropsychiatric conditions, the failure rate skyrockets to nearly 96%. This high rate of failure is largely attributed to the use of animal models in early-stage testing. By utilizing organoids that closely mimic human brain development, it stands to reason that they could become more reliable subjects for developing effective therapies. Conditions like schizophrenia, autism, and Alzheimer's affect the entire brain rather than a single area. By understanding the early developmental issues, we may identify new targets for drug screening, Cathuria explained. We have the opportunity to evaluate new medications or treatment options on the organoids to see if they actually produce the intended effects. This groundbreaking work opens up fascinating avenues for future research. But let me ask you, what are your thoughts on using organoids for studying human brain conditions? Do you believe they might lead to breakthroughs in understanding complex neuropsychiatric disorders? Or do you have reservations about their effectiveness? I'd love to hear your opinions in the comments.